Hello students, today we are starting 11.3, which is called the kidney. We are going to start by looking at the excretory systems. So in today's essential understanding, we see that the type of nitrogenous wastes in animals is correlated with evolutionary history and habitat. So our goal today is to try to find those connections. So what is excretion? Let's first define that. That's the removal from the body of the waste products of metabolic activity. So then that means that defecation is not considered part of excretion because feces is undigested food and it's not metabolic waste. There are two key functions that an excretory system performs. The first one is the removal of nitrogenous wastes, which we're going to see in just a moment, that may be toxic to the body in large concentrations. And secondly, removing excess water to maintain a suitable osmolarity within the tissues and the cells. So let's look and see how nitrogenous wastes are removed. So in this diagram here, we see that nitrogenous waste products vary depending on the organism, as you see here. So these nitrogenous waste products are metabolic waste products from the processing of compounds that contain nitrogen, like amino acids or nitrogenous bases. But depending on the habitat, the environment of the organism, and also the evolutionary history of the organism, you see that the waste product that they release can vary. So for example, most aquatic animals, including bony fishes, release ammonia. Now, ammonia is highly toxic, but this is not a problem for these aquatic animals because they can flush out that ammonia because they live in water and they can take large amounts of water into their body, and therefore that is possible for them to do. Urea is somewhat toxic, not as toxic as ammonia. And you see that the waste product of mammals and amphibians and sharks and some bony fishes, it's urea. And this is because these animals do have some access to water. And this is one way that the urea being toxic can be flushed out, but it's of course less toxic than ammonia. And we see that birds, reptiles, insects, and snails, they eliminate their nitrogen waste products in the form of uric acid. Now this is going to require more energy to do so, but this is okay for them because uric acid is non-toxic or relatively non-toxic. So this is kind of like a trade-off, right? So if you don't have access to water, you have to find a waste product form that is not toxic, right? If the trade-off, of course, is that it takes energy to convert these products into uric acid. But if you live in areas where you have access to water, then you're not going to spend that extra energy. You're going to deal with the highly toxic waste product because you can flush it out with water. So it's kind of like a trade-off, right? All right, so continuing, we see that all animals are either osmoregulators or osmoconformers. So this is the way that animals are able to release or remove the excess water. So a little review here is that water is produced via condensation reactions, and we call that anabolism, and is consumed during hydrolysis reactions, which we called catabolism. Now, the concentration of water within the cells is what we call osmolarity, and this is going to impact tissue viability. So remember when we talked about hypertonic, hypotonic, or isotonic solutions, depending on the type of cell and in what concentration you place them in, that is going to affect the viability of that cell, whether that cell can withstand that environment. So we see here that animals are either osmoconformers or osmoregulators. And that depends on how they are able to manage their internal osmotic conditions. So the osmoconformers right here, they maintain internal conditions that are equal to the osmolarity of their environment. So they don't really fight it, right? If the environment is a particular osmolarity, then they're that osmolarity as well. So they adjust to that environment. Osmoregulators, on the other hand, they keep their body's osmolarity constant, regardless of the environmental conditions. So we see that most primitive organisms, and this is the connection to evolution, right? So most primitive organisms that have a long evolutionary history are mostly osmoconformers. They didn't have the ability for that homeostatic control. As organisms evolved, they were able to apply certain mechanisms to be able to keep their body's osmolarity constant. So more advanced species are the osmoregulators. So in this graph that you see here, we see the difference between the osmoconformers versus the osmoregulators. Now remember one thing about the osmoregulators, right? This is a more energy intensive process. So in order to do that, that's going to require lots of energy. So here we see on the y-axis the internal osmolarity, on the x-axis the external osmolarity. So notice that an osmoconformer, like a jellyfish, is going to change its osmolarity according to the environment, whereas the osmoregulator, for example, the mammal, it's going to keep its osmolarity constant despite any changes in the external osmolarity of the environment. Okay, so that is the initial information that you are required to understand of 11.3, and that is it for today.